SARC Talk. SARC is a nonprofit cancer research organization that develops and manages clinical research trials in pediatric and adult sarcomas. SARC Talk is a podcast to discuss activities of SARC, interview sarcoma research leaders, and review scientific initiatives in the sarcoma field.
Welcome to SARC Talk, episode 35. My name is Scott Okuno, a medical oncologist at Mayo Clinic and chief medical officer at SARC. At SARC Talk, we interview interesting people who are connected to sarcoma, where we can all share and spread the knowledge about sarcoma. Today, we have two guests with us today. Our first guest is Dr. Sandra D'Angelo. She is now a new professor of medicine at Memorial Sloan Kettering as of October of 2025. Congratulations. And we also have Dr. Andrea Napolitano. He is a consultant at the Royal Marsden Hospital in London, England. And they're here to discuss their study SARC 045 for Clear Cell Sarcoma, but also to talk a little bit of how this study was developed, and more importantly, how is this something that we can learn from for future studies. So with that, please welcome Dr. D'Angelo and Dr. Napolitano. Thank you guys for coming tonight.
Thank you very much for the invitation.
Thanks so much for having me back on SARC Talk. It's always fun conversation.
That's correct. And to that point, Dr. D'Angelo, you were here with us on episode 23, where you discuss the role of sarcoma immunotherapy, as well as your publication on the T-cell receptor treatment for synovial sarcoma. And I think that study kind of set the framework because we're the first solid tumor for which T-cell receptor gene therapy has been approved. And I think we still are. So this is great news. So maybe it'd be worthwhile, Sandra, to kind of let people know a little bit about yourself and how you got in the career of immunology and sarcoma, because I know we went over that a little bit in SARC 23 during our last episode 23. But for those that did not listen or forgot, maybe you can share.
Yeah, sure. So I'm a lifelong New Yorker. I joke that I did all my training in different boroughs of New York, but have been here all of my life. I started my medical school training at SUNY Downstate in Brooklyn, and then went on to do my residency at NYU. And actually, I fell into Memorial Sloan Kettering, my first rotation of internship. I was asked to go to Memorial Sloan Kettering, and I covered the inpatient night float service for 2 weeks, which actually started on my 26th birthday, which was a very traumatic 2 weeks. And after those 2 weeks, I said, I'm never coming back to Memorial ever again. This place is nuts. But the work that was ongoing there, the research, the patients, the innovation, obviously allowed me to gravitate and go back. And during my fellowship training, I had initially focused my clinical research in thoracic. When I finished my training, there was no job opening at MSK in thoracic, but there had been a departure on the melanoma sarcoma service. And so I was offered a position, and I sort of fell into sarcoma. And then I kind of fell into immunotherapy as well. Our services later divorced. Melanoma divorced from sarcoma with Gary Schwartz's departure. And I gravitated to sarcoma under Bill Tapp's leadership. But I also became interested in immunotherapy. Jed Walchuk at the time was running an early phase immunotherapy clinic. And Jed said, Sandra, why don't you come do some immunotherapy with us? And so it was really that bridge of immunotherapy with sarcoma, combining, two outstanding mentors with Bill and Jed that introduced me to this field of immunotherapy. And sort of the rest is history. It's just moments of having opportunities and seizing those opportunities that have allowed us to develop these programs at MSK.
Sandra, that is exciting, because a lot of us think we got to have the path marked out for us when you're a resident or a college student and or fellow and or come on staff. And it's kind of nice to say is that, hey, it's okay to work through the path here and to figure it all out. And yeah, maybe there's another detour somewhere along in your life. I don't know, but we're just excited that you're here today to share what you know about the study here, as well as your expertise in sarcoma. So Dr. Napolitano, your training is a little different, partly because you train not in the United States per se, but maybe you can share with our audience here a little bit about your pathway to sarcoma and in the training that you got to the place you are today.
Yeah, sure. Well, I completed my medical schooling in Rome, in Italy, where I'm originally from. And after med school, I actually did end up in the US, but in a very small island. I completed my PhD at University of Hawaii at Manoa in Honolulu, where I studied a germline predisposition to malignant mesothelioma. So that was quite an experience and very interesting 4 years of my life, which really have led me to appreciate even more preclinical and translational research. And after my PhD, I returned back to Rome for my medical oncology residency at Campus Biomedico University. And that center was and is a regional reference center for soft tissue sarcomas. And that's where I had my first experience with sarcoma patients. And as we all know, sarcomas pose unique challenges because of the rarity of the disease and the type of patients that we see. And I just found the combination of the scientific and clinical complexity really, really interesting. And during the last year of my residency, I moved to London as a research fellow in the sarcoma unit at the Royal Marsden Hospital under the leadership of Professor Jones. And after completing this fellowship, I decided to stay in London, where I was offered a job as a consultant. And here we are.
Well, that's interesting. Sandra said she never left the boroughs of New York. You span the countries, institutions. It's quite amazing. So it's fun to see people's career paths. And as you said, when you went and did your PhD, it kind of changed how you thought about things. And then your exposure to sarcoma and then working with Dr. Robin Jones kind of changed your career path. And that's where you are now at the Royal Marsden. So this is great.
Exactly.
So to that point, I'm trying to understand a little bit how you both developed this study together. I know we had a different colleague too that was here that I think he changed out a little bit, but maybe you're working across the pond, as we say, at two different institutions, and maybe you can share with our audience how this study was developed and how you guys collaborated for this.
Yeah, so fundamentally, I think that in sarcomas, we are all used to the idea of repurposing drugs which are being developed for different cancers. So I think that this is a key recurring element in our field. And around the late 2020, early 2021, I think that the data on tebentafusp, this new form of immunotherapy, became more widely available and with really exciting results in another very rare cancer, which is uveal melanoma. And we'll talk more about the details, but to cut a long story short, clear cell sarcoma, they have similar biological characteristics to melanomas. And I think that independently, and that was quite funny, around the same time, people in both institutions came up with the same idea and just said, okay, if this drug really works so well in this rare type of melanoma, maybe there is a rationale for it working in clear cell sarcoma as well. So we really found out about each other when the company that we approached, who has this drug, fundamentally told us, guys, great idea, but you know what? There is another team that fundamentally had the same idea. And so it was just natural to try and partner together as opposed to try this endeavor on our own, considering the rarity of the disease.
It's interesting because, you talk about repurposing drugs. And, Sandra talked a little bit about the divorce we had with melanoma and sarcoma, which is kind of interesting because now we're kind of either be coming in the fellows again together. But to that point, we started sarcoma because of repurposing a drug for gastrointestinal stromal tumor, imatinib. That was originally designed for CML, chronic myelogenous leukemia. We also used pazopanib when it was originally designed for kidney cancer. And then our hope and goal is this drug that you guys are developing for clear cell sarcoma will be another active agent that has been repurposed from melanoma to this. So maybe Sandra, if you are willing, can you share a little bit about what is clear cell sarcoma and why is there some reactivity? If it would work in uveal melanoma, why would it be working in clear cell sarcoma?
Yeah, sure. So clear cell sarcoma is a rare aggressive cancer. It typically forms as a slow-growing mass under the skin. It can start in the extremities, legs, hands, feet, near muscles and tendons. It often affects young adults between the ages of 20 and 40. Biologically, it has a specific chromosomal genetic change, the EWSR1 ATF1 gene fusion, and unfortunately, it recurs and can spread and metastasize to lung, bones, lymph nodes. While there's this connection to melanoma, we now know that it's a biologically distinct and unique disease. What ties tebentafusp to both of the diseases is expression of the GP100 protein. And tebentafusp is a drug that targets both CD3 and the GP100 protein. It's a bispecific antibody. And really, it's that expression of that particular protein that links or makes a connection between the two diseases. But short of that, these are two distinct entities. In fact, that genetic translocation is often what helps distinguish clear cell sarcoma from melanoma.
Absolutely. You brought up a point. Some of us that are listening used to call this amelanotic melanoma of soft parts. And, that might be a little bit of a misnomer, but we do knew there was some type of immunology aspect of this thing here. So this is, quite interesting. So it's a distinct entity. It is not melanoma, and it has a specific translocation. And maybe you can talk a little bit, maybe Andrea, about the actual drug and its targeting. You talk bispecific and a little bit more about that and why you think it's going to work in clear cell sarcoma.
Yeah, so the tebentafusp is a relatively novel form of immunotherapy. So it's a bispecific antibody, which actually a bispecific T-cell engager, which means that it has two portions. One portion which binds T-cells, which is the one that's under the CD3 receptor, which Sandra mentioned. And the other one is binding the GP100, but only a very small segment, a small fraction of the GP100 when this molecule is expressed on a different molecule, on the surface protein, which is called HLA or major histocompatibility complex. This is really key to the activity of the drug because it means that tebentafusp only works in patients that have a specific HLA subtype, which is the HLA-A2. And HLAs are different in every individual, but like we could say similar to a blood subtype. So not all patients will express the right HLA, and tebentafusp will only work in those with the HLA-A2. When a patient has an HLA-A2 and the tumor is expressing the GP100, what tebentafusp does is fundamentally shifts and redirect T cells from the circulation and the microenvironment inside the tumor. And the idea is that this will promote infiltration of immune cells and destruction of the tumor in a disease which is normally, for all we know, not particularly sensitive to immunotherapies, which is the case for many sarcoma subtypes. And I think that the work that Sandra has done with the TCR T cells already has shown that if you have a sarcoma subtype which is not intrinsically very sensitive to immunotherapies, but you can bring the T cells to recognize it, there is a good chance that something positive will happen. And this is what we hope to achieve with tebentafusp.
Oh, that was actually very clear on this as well, because as in the T cell receptor study, you need two things all the time. Just like this study for clear cell, you need two things. You need the right HLA, A2, and then you also need to have expression of GP 100. Is that what I'm hearing? And if you don't have either, the likelihood of this targeted therapy to be effective is not going to be there. So part of this would be is that if you are a patient that has clear cell sarcoma and this study is now available, how do we get patients to the sites across the pond as well as the United States to enroll in this study? So I know, Sandra, you've maybe started this up a little bit. Your site is up and running now, but can maybe describe kind of the rollout plan for sites for this study?
Yeah, sure. So I think one of the most important things is screening for the specific HLA. It can be done in the context of the clinical trial, but that does require a visit at the respective sites that are enrolling. And so we have a screening protocol where patients can sign their consent, and then we send their blood off to be tested for the specific HLA. Alternatively, if this can be done locally at the respective centers, it can maybe mitigate one extra visit if you don't qualify. The limitation of that is that this test may not necessarily be covered. And so it is a little bit of a balance, weighing the cost and travel burden of going to one of our sites that are open and enrolling versus trying to obtain this HLA blood test locally close to home. So once we know that the patient has HLA, if it's been proven already, we've really tried to optimize the process where we can just meet the patient and potentially consent them on the first day of visit at MSK if we know that the patient already has the correct HLA. So I find that that's been helpful to our patients because it's limited travel to New York if not necessary. Obviously, we're always open to meet patients at any point in their treatment journey. But in the context of this specific trial, given the need for that specific requirement with the HLA, optimizing the referral process is probably important. There are other sites in the United States that will be opening. We try to regionally have this available throughout the United States. Dr. Agulnik on the West Coast is almost ready to open. And then I believe at Northwestern, we're also going to be working with Dr. Pollock as well. And so that allows some regional distribution of centers throughout the United States.
Absolutely. And then Dr. Napolitano, how about in Royal Marsden? The plan is also to get your site up and running as well.
Absolutely. The difference is that the regulatory aspects in the UK are slightly more complex than the US. So we're finalizing the discussions with our health authorities, but we hope to be opening within the next couple of months as well. And in the UK, we won't be the only site, but we are quite used to have the Royal Marsden as the main site for clinical trials. So patients are willing to travel. And of course, the distances are not as significant as in the US.
Got it. You know, when we talked in the past for the T cell receptor study in Synovia, you just got one dose. This study, as you know, does require weekly infusions and a 21-day cycle. So there's a commitment that's going to need to be done. And I know your team has worked hard to try to regionalize, make it comfortable for people to come back and forth to a site. So as Sandra, you said too, you're trying to get across the United States. So somewhere in New York, somewhere in the middle of the US, somewhere over in the West Coast, and then clearly in London as well. If people are interested, the site, it sounds like the first site would be, as this recording today, pre-holidays in December, would be at the Memorial Sloan Kettering, and they just have to contact that. That information is available on the SARC website as well. This is a rare tumor, and it does make sense that expertise in developing centers that have, this very unique approach to treatment makes sense. In order for us to get the word out, there are patient advocacy groups, and there is a patient advocacy for clear cell sarcoma. And maybe you can share, how have you used or engaged that group as you developed and continue to recruit patients for this study?
I think partnering with our patient support groups is very important and really an amazing opportunity to allow patients to have sort of more insight and quicker access and really streamlining processes. And so I'm excited about the opportunity to work with the group and really, collaborate to empower our patients to consider this trial as an opportunity in their treatment journey, essentially.
Yeah, absolutely. And I don't know, do you have a similar support patient advocacy group in England as well?
Yeah, there is a group of researchers and clinicians interested in clear cell sarcoma, and we also have the support of the Edward Schauler Foundation specifically for clear cell sarcoma patients. So we are engaging with them and discussing both support in terms of for patients for participating in the trial, also potential ways to collaborate in terms of research with the samples that will be collected during the study. So I think I'd agree with Sandra. Patient support is absolutely fundamental, also, especially in the context of an ultra rare disease like clear cell.
Yeah. Yeah, this is definitely an ultra rare disease for which we are trying to repurpose drugs, so which is a great thing. And, there are many opportunities to do clinical trials. And you guys have been involved with SARC, and maybe you can share how the relationship and how you all decided to run and work together and collaborative with SARC to do this trial.
Like the collaboration with the support groups, I think collaborating with SARC adds significant value and opportunities for investigators like myself and Andrea. I think that SARC brings together a unique opportunity in that all of the centers are all sarcoma centers of excellence. There's tremendous expertise within the SARC community. Further, there's the opportunity to have mentorship, collaboration, translational efforts. And importantly, it also allows us to build out the protocol, the centers, the sites, in a really scientifically robust and rational way. And so it's a wonderful way to really collaborate with my sarcoma colleagues, really with folks that are honed in on the disease and really understand the disease. So it makes the process a little bit more sort of seamless. And I use the word collaborative a number of times, but it's really the perfect word to highlight how SARC functions.
Yeah. We appreciate, the opportunity to work with you on this study. I think it's important. It's harder for us to work across the pond, as we'd say, but, we do have engagement. And we're not the first time that we've worked together. Andrea, maybe you can share a little bit of kind of what you've experienced trying to work through SARC as we've continued to evolve and develop this study.
Yeah, I think that the idea, which is a wrong idea, is that SARC is primarily, a US-based organization and support is primarily for US centers, whereas I found it really, really helpful and they've been great in supporting also on the UK side. And I think that the internationalization of SARC and opening up to expert sites on a global scale is really something important going forward, specifically for, as we said, for ultra-rare diseases where any individual site or even any individual country is not going to have a large enough population to develop significant data. So my experience with them has been great, and they, centralized oversight, experience with regulatory agencies, and support all the way through. So definitely recommending, and I do hope that to have more SARC-sponsored studies in the future in the UK and in Europe.
Okay, that is actually then begs the question. We assume that this study is going to accrue. We're going to find some positive results, although we don't know. What are your next plans? Because, we're always have to think is this took years to develop. We want to know, like, what are you guys working on for future ideas of collaboration and or not necessarily in the clear cell space, but also in the bispecific antibody space?
I think there's a number of different bispecifics that have entered our clinical world. One that I'm excited about at the moment is a bispecific antibody targeting NY-ESO. We know that NY-ESO is a promising target in synovial sarcoma and myxoid round cell liposarcomas. Using bispecifics offers an alternative way and off-the-shelf approach. Mitigates the need for lymphodepletion chemotherapy. And so I think that bispecific antibodies as an alternative or complement to cell therapy will be an emerging field. Further, bispecific antibodies also offer the opportunity to target different pathways. There are a number of novel agents that are being developed. For example, there's a bispecific antibody targeting VEGF and PD-L1 that really has proven to be promising in non-small cell lung cancer. In immunologically cold non-small cell lung cancers, for example, those that have EGFR mutations. And so this is a target that I'm excited about. There's also been some work done with the target of IL-2 and CTLA-4. And so these are sort of interesting and promising drugs that are really kind of entering our therapeutic world, and ones that I envision will be here to stay in the future.
Sounds exciting. Andrea, anything else from your standpoint?
Well, I think that if we look at the future, demonstrating that there are some antigens that can be used to trigger immunological responses in sarcomas, which may be otherwise called, is going to be really, really important also. If we think about, for example, anti-cancer vaccines in the adjuvant setting or in the advanced setting in combination with CAR-T, TCR-T, and bispecific antibodies. So in a way, going back to the clear cell sarcoma, I'm really excited to see if we can show also some sort of proof of concept that targeting melanoma-associated antigen or melanocytic antigens in clear cell sarcoma is something that really provides benefit, because in a way that can showcase more in general that targeting those antigens, even with vaccines, could be an avenue in the future. So that's really what I hope that will be born out of this study.
Well, it's interesting, when some of us that are listening again, it sounded like I'm an old grandpa sitting here thinking about it. The only thing we used to have is two drugs, doxorubicin, maybe dacarbazine, and ifosfamide. And that's when we tried to kill things. And you can imagine the technology, the evolution of what you guys are developing. It's really exciting. And yes, it's an ultra rare tumor, clear cell sarcoma, but SARC-45, I think, will set the tone. We set the tone with the first study, that T cell receptor that Sandra kind of talked about at our last episode. I think sarcoma is going to be a very unique space, and we look forward to what we can develop and continue to pursue and study for our patients in 2026. As we come to a close here, we always want to give you guys the opportunity to say anything you want. But if you were here and you had to be in the elevator, what would be the phrase you want people to remember about clear cell sarcoma or SARC-45? I'll start with you, Sandra.
Well, I mean, I think if you have a patient with clear cell, I would advocate for screening for HLA. Communicate with the investigators that have the trial open, and really just think about this opportunity as an alternative way to treat this cancer.
Perfect. So Sandra listens pretty good. So that's more than 3 words. That's more than a phrase, but it's pretty good. But it's really important, unless you test for HLA and or the GP100, you're never going to get into that space. So if you are out there and you have advanced sarcoma, clear cell, you just, part of the reason why we're sharing this is to get the word out that, yeah, your site should test for it. If not, as Sandra said, get to a site that can screen for you for this potential study. Andrea?
The 3 words that I thought when thinking about Clear Cell and SARC-45 in particular are hope, precision, and collaboration. And I think that these are the key elements of this study.
I love it. Hope, precision, collaboration. We got to write that down. I'm going to have to remember that. So this is all good. Are there any thoughts that you have? And I do appreciate the time that you've both shared to develop this study and continue to work together in hope, precision, and collaboration. But any other final thoughts?
No, I have to give kudos to Andrea for highlighting those 3 words. Task I wasn't able to do, but it's again, this is just a great forum to share research, our ideas. I also just want to reiterate the collaboration with Andrea and the Royal Marsden, and really uniting us across the pond is really a powerful tool, and using SARC as a mechanism to do that can really maybe lay the groundwork for future efforts and future trials. Many of us always have the same great ideas, and really we don't want that to be a barrier. We want it to be a bridge to really bring promising therapeutics to our patients. And so this has been a really nice journey. And I do have to call out Evan Rosenbaum, who really initiated this initial work and protocol drafting, and really formed the foundation for the collaboration.
No, I do. Yeah, I forgot. And, Evan worked very extensively with you, Sandra, there to develop this study. And if he's listening, we thank him for all his efforts and collaboration in developing the study as well. Andrea, anything else?
Not really. I just want to thank you for having me in having this chat. It's been really, really interesting. And again, just to reiterate what Sandra said, that we hope that this is really going to be a new treatment opportunity for patients, but also the beginning of a collaboration with SARC more in general to bring new therapies for rare and ultra-rare sarcomas involving centers globally.
Perfect. So thank you both. Thank you both for your expertise and time today. And for those that are listening, we hope when this gets published that it'll be a brand new 2026. And if you have any comments or questions, please post them along the way, because we do listen and review those, and wishing for a great and successful completion of this study and continued collaboration. And we look forward to 2026. Thanks, everybody.
Thank you.
Thanks so much, Scott.
Thank you for listening to SARC Talk. Be sure to subscribe so you never miss an episode. To learn more about SARC, visit us at sarctrials.org. If you have questions or would like to suggest a topic, email us at sarctalk@sarctrials.org. SARC Talk is made possible through the generous support of Surier Pharmaceuticals. Together, we can find a cure for all forms of sarcoma.